Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about microscopy, introduction to microscopy. We're going to talk about the objectives of the overall uh, uh, talk on microscopy, talk about magnification, resolution, and we'll be covering some blood cells as we look at using the different types of microscopy. We want to look at differences in magnification and resolution, to learn a little bit about different types of staining at light microscopic level and some things about details that we see at the electron microscopic level. Learn how cell and organelles look different at different magnifications at the electron microscopic level, and to learn about differences in different types of electron microscopic images. These are the four basic specimens that we'll be looking at today as we talk about microscopy. One is the pancreas, and we can see pancreatic astrocells that produce pancreatic enzymes. And then here the lighter staining are the islet cell Langerhans that produces insulin. Here we see blood cells of different type. There's eosinophil, neutrophil, red blood cells, different blood cells that we'll see. And here is the mucosa of the stomach. And here we see the parietal cells, and there's some chief cells in there as well. And then we'll end up with the testis, showing you the tubes, the cells in the tubes, and then the interstitial cells between the Leydig cells in between. Just wanted to say that these are atlas views, and it'd be very handy if you would use your atlas to be able to observe these structures, because indeed the structures themselves are labeled so you can identify the various components that you're looking for. Now, what's the difference in magnification and resolution? Now, magnification is simply the increase in the image size. Just keep increasing in size, but if you do that, you will go into empty magnification, namely that you will not gain anything by increasing the image size. Resolution is the important part about microscopy. It's the smallest distance that can be seen between 2 points. If you ever look down the straight railroad track and you note that as you look, the distance look like the two rails become together and then they come one. You know they don't come together. What you're doing is you're losing resolution with distance. So you can't distinguish between the two rails anymore. That's resolution. And resolution can be calculated by a formula, a constant 0.61 times the wavelength divided by the numerical aperture, or the light gathering capacity of whatever microscope you have. For your naked eye, you usually see things in millimeters, but for a light microscope, it's micrometers that we see. 0.25 micrometers is your resolution. The resolution for electron microscope is nanometers. So we go from millimeters and micrometers to nanometers in terms of much finer detail can be seen with the electron microscope. And the reason the resolution is so much smaller with the electron microscope is because of wavelength of light. In the electron microscope, we use electrons. Electron is much smaller than white light. As a consequence, you have a smaller number at the very top, and so the resolution is better at the electron microscopic level. Now, as we look at different components of microscopy, we can see one thing that we want to see is of cell, blood cells. This is individual cell, another individual cell, and by looking at blood cells, you can see individual ones, and don't be confused with the cells in the tissue. This is a eosinophil, monocyte, basophil. Here we see eosinophil, neutrophils, as we see, these are all from the atlas and identified for you. Now, the point of this lab is not to identify the white blood cells yet. We will have a whole lab on blood. But the point here is just to look for different colors and the staining characteristics that facilitates those differences in colors. And basically, the neutrophil, eosinophil, and basophil all have lobulated nucleuses, lobes. You can see different lobes. And then the granules are small in a neutrophil, but eosinophil are bigger red granules. And then the basophil has big granules too, but they're blue. And then the lymphocyte is the smallest one, and the horseshoe-shaped one is the monocyte that we can see. Basically, that's it. And electron microscopic level, you can see are some granules, but they're smaller in the neutrophil than they are in the eosinophil and the basophil. So these two have larger granules. This is eosinophil. It has a core, a crystalline core in this granule that you don't see in the basophil. And then we don't see any granules. We just see mitochondria. And then here is a monocyte. So if we look at one of our blood samples, we see many of these cells are red blood cells, and the little dots in there are the platelets. So here we see a monocyte, we see there's a lymphocyte here and here, but that's a neutrophil that we see. We look at this one, 110, we can see a neutrophil, which you can hardly see the granules, and then you can see the eosinophil with their big red granules. Both of these are a lobulated nucleus, and both of them about 12 to 15 micrometers in size. So here we see more neutrophils, and here's a basophil, where the lobulated nucleus and blue granules instead of more neutrophils, red blood cells, as we can see. Here we see the platelets, nicely staining platelets, and the red blood cells are stacked up and through there, and the neutrophils. In a neutrophil, you don't see the granules, you just see a granularity of it because the granules are not that large. And in fact, if you look at a neutrophil electron microscopic level, you can see the different lobes of the nucleus. So this is a nucleus, and then these are the granules that you can see, and these neutrophils that we see through here. And so these are different ones, are neutrophils. I can't really see mitochondria in through here, but you can see the granules. See, this monocyte doesn't have the granules that you see in the neutrophil. We want to thank the original source of illustrations, pictures, and drawings that were used in this presentations that came from original books, and we herein acknowledge their contribution. So this is the end of the introduction to microscopy, the objectives of the overall talk on microscopy, magnification, resolution, and blood cells. Thank you.